Hi, everyone. Welcome to the 2025 AGM Preclinical Translational Science webinar series. For anyone that does not know me, I'm Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences. We're really excited to have you all here with us today, and we're actually super excited to have our first speaker. I'm super excited to introduce our first speaker for this year, who is Dr. Katherine Hai. Dr. Hai's career has spent over 35 years in internal medicine, hematology, molecular genetics, and gene therapy. Her pioneering bench-to-bedside studies of gene therapy for hemophilia led to a series of basic and clinical investigations that characterized the human immune response to AAV gene delivery vectors. She's a longtime member of the faculty of Perelman School of Medicine of the University of Pennsylvania. She was also an HHMI investigator at the Children's Hospital of Philadelphia. In 2013, Kathy co-founded Spark Therapeutics, where she served as president, CSO, and member of the board of directors. At Spark, she led the team that achieved the first FDA approval of a gene therapy for genetic disease Luxturna for a rare form of inherited blindness, and also led the successful phase 1-2 studies for a gene therapy for hemophilia B, now approved product following a Pfizer-sponsored phase 3 study. In January 2021, she joined AskBio as president of therapeutics and member of the board of the directors. And in 2022, she left AskBio. She's currently a visiting professor at Rockefeller University. She remains a professor emerita at the University of Pennsylvania School of Medicine. And also just recently, she became the CEO of Rigase, a biotech startup located in both Basel and in Philadelphia. She's an elected member of the National Academy of Medicine, the American Academy of Arts and Sciences, and the National Academy of Sciences. And today she's going to talk to us about the relationship of preclinical studies to clinical development in AAV gene therapy for genetic disease. Kathy, we are super excited to have you here, and we look forward to your talk. The floor is yours.
Okay, thank you very much. You can hear me, right?
Yes.
Okay, great. So I'm actually really glad to be here. I'm glad that these activities are continuing. And I'm just going to start by these disclosures, which I think were mostly already discussed. What I'm going to talk about here today is mostly the inherited retinal dystrophy trial, because I couldn't talk about everything. But I wanted to talk about two studies, the farm tox studies that guided the selection of the starting dose, which had to be interleaved with requirements for the inclusion of children in investigational studies because, and I'll show you why, because the preclinical data in a dog model of this type of blindness indicated that the earlier the patient underwent an intervention, the more likely they were to have a positive outcome. And then secondly, the work that went into developing and validating the potency assay for the vector. Now, I'm going to assume that everybody in the audience knows a certain amount about AAV vectors. And if that's a bad assumption, I can walk you through a little bit without a slide. But let me just say a little bit about this rare inherited retinal dystrophy. It's actually an ultra-rare condition, but it's an autosomal recessive disease caused by a mutation in a gene with the unwieldy name retinal pigment epithelium 65 kilodalton protein. And these individuals typically present fairly early in life with visual impairment. And for most of them, it progresses to blindness, certainly, if not in the first decade of life, into the second decade. And before this work, there was no pharmacologic means of treating the disease, but there were genetically engineered mice and naturally occurring dog models of the disease. And just to orient everybody from an anatomic standpoint, the defect is in, I don't know if you can see my cursor, but the retinal pigment epithelial cells, which are shown here in gray, form the blood-retina barrier, and they also carry out much of the metabolism of the photoreceptors, which are some of the most metabolically active cells in the body and cannot complete all of their own metabolism without the help from these retinal pigment epithelial cells. And so from a biochemical standpoint, if you look in this lower portion, the blue portion, when light strikes the retina, you change an 11-cis form of retinol to a trans form, and that starts the action potential down, back through the layers of the retina, back through the optic nerve. But the trans retinol has to be metabolized to regenerate cisretinol. And in order to do that, it's transferred back out of the rod outer segments of the photoreceptors and back into the retinal pigment epithelial cells, where under the action of two enzymes, lecithin retinol acyltransferase and RPE65, 11-cis-retinol is regenerated. And without RPE65, this visual cycle is broken, and eventually you begin to accumulate toxic metabolites that eventually interfere with the metabolism of the RPE cells, and when that happens, that begins to interfere with the photoreceptors, and vision gradually deteriorates. So what Jean Bennett and her colleagues at Cornell, at the University of Florida, and at Penn had shown was that they could supply the missing gene in a subretinal injection of an AAV vector expressing RPE65, and thereby rescue vision in dogs born with this disease. They got very long-lasting expression, but they needed to inject these animals relatively early in life to avoid the period where the vision had already deteriorated to the point where they weren't going to be able to rescue it. I will point out to you that the same is not true for all humans, right? I mean, humans have many different mutations in the RPE65 gene. This colony of dogs all had the same mutation, and they deteriorated on a very fast timeline as these things go. But humans, depending on their mutation, may have vision well into the second decade of life and rescuable vision for longer than that. But anyway, so we wanted to try to listen to this preclinical data and from the very beginning include children in the trial if we could do so safely. So I'll just tell you this, that in the dog model, if an immunofluorescent stain for RPE65, you can see robust presence of the protein here in a normal dog, and its absence in the dogs born with a mutation in RPE65, and that the administration of the AAV vector in the dogs restores the presence of the missing protein. And so that was what we knew when we began to work on the clinical trial. And so what we needed to do was translate what we knew about expression and doses into a rationale for the correct starting dose. And so what we had originally proposed was to start at a dose of 1.5 times 10 to the 9th vector genomes of the vector and dose escalate in 1 log steps, 1.5 E9, 1.5 E10, 1.5 E11. This proposed starting dose, we knew, restored vision occasionally in affected dogs, although most dogs did not regain vision at that dose. And in our discussions with, in our initial discussions with the RAC and subsequently with the FDA and the EMA, that dose was approved as the starting dose. However, and again, we knew that at doses beginning at 1.5E10, it restored visual behavior in the dogs, in most all dogs that were injected. So the issue arose when we began to talk about including children. And if you, the inclusion of children in as research subjects is governed by a particular part of the Code of Federal Regulations. And what it says basically is that children can participate in research involving greater than minimal risk, as long as there's the prospect of direct benefit, and that they can participate in research that involves more than minimal risk and no prospect of benefit, if the IRB finds that the risk represents a minor increase over minimal risk. And so when we discuss that with, and I won't go through the federal minimal risk standards, but essentially minimal risk is what's encountered in the life of an average healthy child. And so this administration of an AAV vector to the subretinal space does involve general anesthesia, the risk of which is relatively low in a healthy child. And so when the RAC assessed this, they looked at the risks of the surgery, the risk of undergoing general anesthesia, the risk that the vector might be distributed beyond the site where it was injected, the risk that it might result in inflammation, and that might somehow diminish the visual function in the treated eye. But they still assessed all these risks as slightly greater than the minimum risk, although they did note that only the institutional review board at the hospital is responsible for the risk-benefit assessment. They felt that this was reasonable, that it represented only a slightly greater than minimal risk. But when the Children's Hospital of Philadelphia IRB assessed that, they said that this study did involve more than minimal risk, which I think it would be difficult to argue with for undergoing a surgical procedure and experiencing general anesthesia. And so we had to revisit the data. We had done additional experiments at Spark to quantify the proportion of normal RPE65 activity following an injection of 4 × 10 to the 10th vector genomes in the eye. And we had quantified that as driving about 55% of normal levels of RPE65 in a dog born without any RPE65 expression. And so we also assumed that a dose of 1.5E10, which we knew restored visual behavior in these dogs, and would therefore, we extrapolated, express about 20% of normal RPE65. So we made that the starting dose. We proposed that using all the safety data we had to the FDA in a resubmission, and they agreed that that was an acceptable starting dose. And so we changed the dose escalation to half logs, 1.5E10, 4.8E10, and 1.5E11 as our dose escalation. And that was approved by both the CHOP IRB and the FDA. And we were able to begin. We also looked at what we would use as the upper limit for dose escalation. And again, in the dog model, at a dose of 1E12 vector genomes per eye, we started to see focal ocular pathology. So we did feel that the no observed adverse event level was going to be lower than that. In non-human primate studies, both 3E11 and 7.5E11 were safe. There was no dose-limiting toxicity. But again, we felt that we were getting adequate expression at 1.5E11 based on everything we knew from studies in the dogs. So as I mentioned, our dose escalation doses for the human eye are listed here. So we looked at 3 different doses. We saw one adverse event in the mid dose. The patient developed bacterial endophthalmitis after the procedure. And he had been given periocular steroids as a routine part of the surgery, just to try to reduce inflammation. And then he developed increased intraocular pressure in response to those, along with the bacterial endophthalmitis, for which he was treated. But anyway, other than that, we did not see evidence of inflammation at the 3 doses tested. And I'll just add that another trial, which did advance to 1E12 vector genomes per eye, did see inflammation and macular thinning in multiple patients who received that dose. So we felt that, somewhere between 1.5E11 and 1E12 is probably a maximum dose that can be given, but we felt that based on our animal data, we were expressing adequate levels at 1.5E11. So that was the dose that we moved forward with in phase 3. Okay, so I wanted to talk a little bit now about the potency assay that was developed for the vector itself as part of lot release criteria. And let me just say that when we had begun these studies, and I didn't say this at the beginning, but we began all these studies as studies at the Children's Hospital of Philadelphia, sponsored by our unit at Children's Hospital of Philadelphia, the Center for Cellular and Molecular Therapeutics. And we transitioned that eventually to Spark Therapeutics during phase 3. But while we were at CHOP, we were using a potency assay that was actually an in vivo study in a mouse model with blindness. So let me say, first of all, that the 2 key goals in a potency assay for an AAV gene therapy product are to answer, first, how well does the vector transduce target cells? And secondly, what's the level of activity of the transgene product? This mirroring assay was, first of all, pretty slow. We injected mice early in life with our vector, and then looked 6 weeks later to see if the mice who had been born blind had developed a pupillary light reflex. So obviously, this was a qualitative assay. Either they had the pupillary light reflex or they did not. And the regulatory agencies had urged us as an early step to move to a biochemical assay that would be more quantitative. And so we made attempts to do this at CHOP, and we were not successful. But when we had a larger scientific staff at Spark Therapeutics, we were able to do this. So, I'm just going to show you this part of the visual cycle again. This is flipped from what you saw before. The photoreceptors are up here, and the RPE cells are down here. But just to remind you, the transretinal that is formed when light strikes the retina and is then transported back to the RPE cells needs both this less than retinol acyltransferase and RPE65 to be regenerated as 11-cis retinol. So we actually selected as our cell line that we were going to do this in HEK293 cells, because we knew that they were readily transduced by AAV, so we could assess that is our vector transducing cells. But our HEK293 cells, which are a human embryonic kidney cell line, don't express these retinal-specific proteins, enzymes, LRAT and RP65. But the scientist who was leading this, Dr. Linda Couto, found a cell line that had been stably transfected with LRAT, this one enzyme that we needed. And so then the concept of this assay was that you transduce the HEK293 LRAT cells with the vector. You lyse the vector. You supply the substrate for RPE65 in excess, all transretinal. You also include this bonding protein that is needed to stabilize the 11-cis retinol. So this CrowBP has to be also supplied because it's not made in 293 cells. And then you can use mass spec to quantify the amount of cis retinol that you've regenerated in the reaction. Now, I'll add this. All of these pigments, all of these compounds are light sensitive. So this reaction from the lysed transduced cells has to be conducted in the dark. And then the reaction is quenched and you quantify the amount of cis retinol that's made. And again, both the CrowBP and the all transretinol are in excess. The limiting factor is the amount of RPE65 produced by your AAV vector, and that's how you quantify how much expression is being driven by the RPE65 cells. Dr. Couto is one of the most amazing scientists I've ever worked with. I have to tell you this just as an example of the problems that were overcome. When she was working up this assay, she found 3 vendors in the United States who made CrowBP. Only one of them made it under GMP conditions. She ordered all 3 and tested them. Only one worked. It was not the GMP reagent. So she had to go to the supplier who manufactured this and ask him if he would begin producing this under GMP conditions. And amazingly for us, his brother had been an ophthalmologist who sadly died in his 40s from cancer. And in honor of his brother, he agreed to do this. I mean, he did charge us a fair price for it, but he agreed to produce it as a GMP reagent, and so that's what we used in this assay. So again, to do this for lot release, we needed to transduce at 9 different multiplicities of infection from a low vector genome per cell to a high one that would allow us to get to an asymptote at the top of the curve. We did each of these multiplicities of infection in 6 replicates for each assay, and then lysed the cells, quantified the amount of protein, and then set up this reaction in the dark. After 2 hours, quenched the reaction, and then did mass spec to quantify the amount of product that was regenerated. Okay, so I think I've gone through everything in this. Yeah. Okay. And so the curves, the mass spec curves, there was at low MOIs, this was a little bit hidden by shoulder, but as you increase the MOI, it was fairly straightforward to see this. And with 9 different MOIs, we were able to develop a pretty good curve with an asymptote at the top of the range. And so in each of these assays, we compared, this is a relative potency assay, so we're comparing the test article to the reference standard. To demonstrate specificity, we did this compared to adding, instead of the reference standard, the formulation buffer. You can see that the formulation buffer has 0 activity without the AAV vector in the reactions. And so it is specific for the presence of AAV RPE65. We showed that the results were similar between 2 different analysts who were carrying out the comparison of the reference standard to the test article, and within a very close level, they agreed with each other. We prepared dilutions of 50% test article, 75%, 100%, and 150%. The test article is in green, the reference standard is in blue, and you can see as you increase the concentration, of the vector, you get closer and closer to a match between the reference article, the reference standard, and the test article, and at 150% you can distinguish these. And we also demonstrated robustness by varying a number of the conditions of the assay. Here we varied the transduction time from 24 hours down to 18 hours and up to 26 hours, and showed that that did not make a difference in the results. We changed the sonication pulses for making the lysates from 2 pulses to either 1 or 3, and that didn't make a difference. And we varied the crowd BP concentration from the set concentration of 25 microliters down to 20 and up to 30, and that didn't make a difference either. So we were able to demonstrate that it was a fairly robust assay. All right, so that thankfully passed muster at the FDA for and the EMA for the potency assay for the material. It's used to release the product to determine the stability of the product over time. Actually, we were able to use the same assay to assess variants of unknown significance by just making those mutations and putting them into this assay. You can assess whether a variant of unknown significance has activity in this reaction or not. So now let me see. Okay, in the last few minutes, what I want to do is just tell you a little bit about the trial itself. This is actually looking through the operating room microscope. This is the lead surgeon, Dr. Albert McGuire, an ophthalmologist at Penn. And what you see when you look through the operating room microscope is how the bleb is raised that contains the vector. Maybe I'll just play that again. So the vitreous is removed in a vitrectomy, and then the surgeon can, under direct visualization, direct the injection to the area which has been delineated ahead of time in imaging procedures. And then this shows the entire clinical development program. There was a dose escalation study as phase 1 first in 12 individuals who all had different mutations in different stages of disease, but it was just really done for safety and to get preliminary evidence of efficacy. All 3 doses were safe, and we therefore chose as the dose to move forward into phase 3 the highest dose because it had the largest volume and covered the greatest amount of the retina. In a second study in phase 1, we went back. The first phase 1 study was dose escalation in the worst eye, the worst of the 2 eyes. In those 12 individuals, we went back and injected the contralateral eye with the dose that was going to be carried forward into phase 3 in all of those individuals to show that it was safe to inject both eyes. And then the phase 3 study was done as a randomized controlled crossover study. I'll say a little bit more about that in a minute. We had to do a natural history study. Most of the data in the literature was single institution case reports. But just to have a point of comparison, we did do a natural history study that involved a retrospective chart review of 70 cases of this disease at referral centers on 3 different continents, which is the largest study of patients with this disease in the literature now. And we did a separate study looking at our new primary endpoint, which was essentially a navigation test, a mobility test, where we vary the level of light in the room because these individuals are sensitive to the amount of light, and at very bright light they may be able to navigate, but as you dim the lights they really can't. So this primary endpoint we had to develop and validate. which I'm not really going to have time to talk about, but just, we published all of this work. This is the phase 3 study in the upper left, and the mobility assay and its performance characteristics in the upper right, and the natural history study in the lower left. So, so in terms of the phase 3 trial design, let me just say this. We discussed at length with the regulators what we could use as a control in the phase 3. As I mentioned at the outset, not very good natural history data. This was one of those building the airplane while you're flying it exercises, and we were doing the natural history study as we were doing phase 3. So we didn't have enough good natural history data to use historical controls. The phase 1 trial was originally done looking at the first eye compared to the uninjected second eye, but the FDA was not enthusiastic about that as a phase 3 trial design, since they felt that no one would be using the product that way. And so what we eventually settled on was a randomized controlled trial design, in which individuals who were deemed eligible were randomized 2-to-1 to either undergo intervention where first the first eye, and then within 1 to 2 weeks the second eye were injected, or to be randomized to the control group, where they underwent all the same evaluations but were uninjected, and then at the end of 1 year they could cross over to the intervention group. And oops, I really need to wrap up here. Let me just say that we could not do sham injections because we had children in the trial. At phase 1, we had children down to the age of 8, which is the age of assent at the Children's Hospital of Philadelphia. That means that you can explain it in age-appropriate language, and the child can either agree or not agree. Obviously, the parents have to give consent. But based on the safety data in phase 1, the regulatory agencies allowed us to include children down to the age of 3 in the phase 3 study. So crossover, yeah, okay. And then at the end of 1 year, all these individuals could cross over to receive injections in both eyes. So I'm not going to go through this material on the this multi-luminance mobility test, but just suffice it to say we
Jump in here. You can take till 12, that's fine. I don't see a lot of questions, so if you would like to take more time.
Okay, well, let me just say then that, let's see if the issue is this, right? We all know how to measure visual function. You go to the eye doctor, you get your visual acuity done. They can do visual fields. They can see how well you can read contrast sensitivity. But the regulators on both sides of the Atlantic wanted a measure of functional vision. That is, how does a person function in a visually dependent activity of daily living? And again, reading is obviously such an activity. So is navigation. And because we had young children as trial participants, we decided it would be better to do a navigation test, a mobility test. These tests do exist, but they were not structured, the ones in the literature were not structured to be dependent on the level of illumination. And so that's why we had to come up with our own test. And what we came up with was a test where the patient needs to follow a series of arrows on the floor that are set up in the ophthalmology clinic. And there are obstacles along the way. So here's a step where you have to step over. You're supposed to avoid things like these black holes, step over another obstacle here, and then get to the end and open the door. So here it is at a bright light level, but it was also done at much dimmer levels of light, all the way down to 1 lux, which is essentially the level of light you see with an indoor night light. And, going up in gradations to brighter and brighter light. And the way it was used in phase 3 is you determine the lowest level of light where the patient can get a passing score. And their passing score has to be, they have to do it quickly enough, although we let them have plenty of time to get through before we failed them on time, and accurately enough, so they can't make mistakes, step on obstacles, and so forth. And then you compare that to how dim light, what level of dim light they can do that in 1 year later. Now we checked at 30 days, 90 days, 180 days, and 365 days. And at the, I'll skip over that. Well, this just shows you how one of these patients did. This is one of the phase 3 patients. And what you're going to see, at baseline, she fails at 1 lux. And at 1 year after treatment, she passes. When I turn this on, you will see the patient moving through the 1 year course pretty rapidly, as if, I mean, really not much worse than any of us would do. And then you'll see that at baseline, she's not able to do it. So the examiner says start. Other than that, he doesn't talk. They don't talk in the exam room. And she steps over the obstacle here. She steps over it here. Okay. And you see she gets to the door and opens the door. Now, at baseline, you see she had already wandered off the course.
Redirect.
The examiner says redirect. That means she gets a penalty for that. Now she wanders off on the other side of the course. And the examiner on that side has to get her back onto the course. She collides with the obstacle on the ground there. And you can see that I think you can see this, that she's, to some extent, she's feeling with her feet. And that is, that's not what she's supposed to be doing. And so even without running it all the way to the end, I think you can see that she failed at the baseline and passed with flying colors at 1 year. So that was how it was used. But all right, I'm going to have to skip over all this. I can't. Let me just tell you the results in phase. I mean, at the end of this, we had 3,800 videos between the validation study and all the patients in phase 3, because we recorded, at each run of the course, and I did skip over that, but we had 12 different courses which were presented in randomized orders to the patient to try to mitigate learning effect. And then, we had very robust statistics on how well all of these videotapes were sent to an outside grading center in shuffled order. So they were just grading videotapes. 2 graders graded each videotape, and if they did not agree, there was an adjudicator who would look at the tapes. But what you can see is that for the individuals randomized to the intervention group, within 30 days, they were now able to pass in the aggregate at 4 lux, and many of them were able to pass at 1 lux, and that they held that for 1 year, whereas the people in the control group who didn't get injected, they may have improved a little bit, but not much. And then at the end of 1 year, when they were all injected, they recapitulated the results from the original control group after they were injected. And so, we had very robust results. We actually over-enrolled in the trial compared to what the statisticians told us that we needed, because I couldn't believe that they were right when they said we only needed 16 patients in the intervention group and 8 patients in the control group. So we over-enrolled, and we ended up with quite robust statistics. Look, I think this is important too. This is in the first year. The intervention group went from 100 seconds to get through the course to 49 seconds. And I think the clinical meaningfulness is obvious to that if you think about crossing the street. Whereas the control group, in the year that they were not injected, essentially they did not change in the time it took them to complete the course. So I'm just going to stop here and say that, again, we over-enrolled in the trial, and we ended up with very robust statistics for the primary endpoint and the first 2 secondary endpoints. We missed on visual acuity, but we had this exploratory endpoint of visual fields where we had very robust statistics, and we should have made that one of the secondary endpoints, but we didn't figure it out based on our data from the small phase 1 study. I mean, that's a pitfall of what we did there. There was no FDA guidance document on inherited retinal dystrophy gene therapy when we began this work, but I am happy to say that in the document that they subsequently wrote, they called out that sponsors were welcome to engage FDA to work on developing acceptable endpoints. And really this endpoint was developed in close dialogue with the FDA. They provided a lot of helpful recommendations to us before we began the trial, including videotaping every run of the course, which later people told us was important for developing convincing data. Okay, I will just conclude by saying that this program in aggregate, not counting the decades of work that Jean Bennett had done before we began working together on this, but still to get to approval took us 12 years of collaborative work. And, you know, all of this was based on work that Jean Bennett had done with her colleagues at Penn, University of Florida, and Cornell Vet School, making very valuable use of the naturally occurring dog model. My colleagues at Spark Therapeutics, the University of Iowa was the second site for the phase 3 study. We had terrific biostatistics help, and ICRO was the group that made the CRAL BP at GMP grade that enabled us to develop a potency assay. So, okay, I'm going to stop there. I know we don't have much time for questions, but Abby, should I stop sharing? What should I do here?
Yes. You can keep your slides. I'm Serene. Yeah, you can keep your slides. And what we can do is we take some of the questions that are here. And if you have time, we can go a little bit over 12. If that's okay.
I have time. I have time.
Great. Awesome. So we have multiple questions. So I'll start with asking a few. So this is from Kapil from NEI. Thanks for sharing Thanks for sharing such a detailed preclinical development and clinical trial data for Luxturna. 2 questions. First, for rare disease populations, if one manufactures just one large lot of the drug product and want to use it all the way into the commercial stage, what time frame is considered sufficient to demonstrate long-term stability of the drug product?
Well, let me take that one first. Don't ask me 2 at once. Okay, so
yeah, I stopped.
In terms of the time frame, my colleagues in commercial manufacturing tell me that it is unusual to date material for longer than 5 years. We began the clinical trial in 2007, and I think the last patient was injected in 2013. Now, we also, I think we're very much encouraged by the FDA to use more than one lot. And so we did do that. So some of the patients were injected with the first lot and some with the second lot. And of course, typically you have to give them data on more lots than that, not clinical data, but manufacturing data on more lots than that so that they can determine that you have a reproducible process. But, you know, we use 2 different lots in the clinical development program, essentially as per their recommendation. And we had more lots than that were submitted, you know, engineering runs and so forth. From the facility that we built at Spark, that was the facility that underwent the pre-approval inspection. Did I answer that?
All right. Thank you so much. Yeah. So the second question is, Slightly more provocative question. The success we saw with Luxturna couldn't easily be replicated with some other inherited retinal degenerative diseases. What are your thoughts about this? Do you think we will continue with this one gene therapy approach, or do you see the field moving towards a more gene agnostic gene therapy approach?
Well, let me say, first of all, that RP65 was a very felicitous choice in a number of ways. First of all, you do get a gain of function. And I think we all recognize that a lot of slowly degenerative conditions, unfortunately, all you may see is a stabilization of the patient where they are rather than a gain of function. If, for example, cells are already beyond the point where they can be resuscitated. So you may save some of the cells, but you can't bring to life cells that are no longer there. And so then you may be looking at very long clinical timelines. If it's a very slow moving disease and you're not getting gain of function from supplying the gene. So I think that's been a challenge for some diseases. And of course, retinal pigment epithelial cells are not only easily transduced, but they actually will eat anything that they find. And so I suspect that you get pretty robust transduction of those cells. And can we get comparable levels of transduction with photoreceptors, human photoreceptors? But to your question of, will it be possible for people to develop generalized solutions for inherited retinal diseases? Well, I certainly hope so, because if you look at the time it took to develop Luxturna, and you think about 350 or more genes involved in human vision, you can readily appreciate that the likelihood of solving this one gene at a time is going to take person centuries of work to accomplish. So I can only say I certainly hope that there will be a more generalized solution, and keep working.
Thank you, Kari. Wonderful. All right, more questions. So I'll go through some of them too. This is from Emily Lee from NCATS, I believe. Thank you. Yep. Thank you for sharing this talk with us. Two questions. Was the same serotype used in each species, dog, NHP, human, or is the serotype adjusted per species?
Well, we actually used, Gene had used AAV2 in the dogs, but all of our work in the dogs, the non-human primates, the mice that we used for the potency assay, and humans, for all of those, we used AAV2. So it was the sort of oldest and best known serotype. And, we didn't change among species.
Okay. And the other question, can you comment on whether there are species tissue-specific differences in AAV transduction efficiency, for example, between dogs and NHPs and humans? And if so, how are these potential differences accounted for when choosing a dose used in the human clinical trials?
Well, that's a really good question. And I would say that a large body of work in a variety of different target tissues would say that these relative performances in different species are not well conserved. So, I mean, we were fortunate with AAV2 in RPE cells that it seemed to translate easily from one species to another. But I mean, for example, AAV8 in the liver, very robust transduction in mice, and in humans, really no difference from AAV2. I mean, logs better in the liver in mice than AAV2, in humans, no difference. And so, this is something that unfortunately has to be worked out in clinical investigation, because one can get a reasonable idea from studies in animals, but it may not be a robust conclusion in moving to clinical trials. There's an advantage here if one knows the receptor, because if you know the receptor, you can actually look at the human cells and culture and say, is it abundant there as it is in non-human primates or whatever species you're comparing it to? But viruses are very clever, and sometimes if they don't see the receptor they're used to, they can adopt something else. And so, even that may not give you the answer. So, I would say for gene therapy, there are a number of aspects that really can only be worked out in clinical investigation.
Thank you so much, Kathy. Okay, so more questions. What was the rationale for using NHPs in the preclinical safety studies in addition to the dog as the relevant species?
Well, that's a really good question. I think that we were eager to develop as robust a package as possible, given the fact that we were asking to include children early on. And that's really, it's a fair question. And I think there were arguments on both sides of using just a large animal model of the disease versus non-human primates. And we just made the decision to do both of them.
Oh, thank you. And there's two. Go ahead.
No, I don't have anything to add to that.
Okay, two more questions from the same person. Were any other factors considered to account for uncertainty in determining the starting safe dose for humans based on the NOAELs from large animal models?
Were any other factors considered? Well, as I tried to indicate, in order to include children, we did have to get to a dose that would reasonably be predicted to provide benefit to each person enrolled. And that was what caused us to increase the starting dose from 1.5E9 to 1.5E10. And I did talk about that. I don't know if you're referring to some other factor.
Yeah, that's what I see in the question. Where any other factors considered to account for uncertainty in determining the starting safe dose for humans based on the NOAELs from large animal models. Okay. So, and the third question here is, were any concerns raised by regulators or the FDA on non-specific integration of AAV prior to starting the phase 1 in humans? And if yes, were any additional test preclinical data requested?
Actually, I don't remember that being raised as an issue. You're probably aware of the fact that the doses used in these ophthalmology trials are relatively low compared to systemic administration. And I think it was certainly raised in trials for hemophilia, for example, that we were involved with, where the dose is logs higher than what is used here. But no, actually, I can't remember. It may have been raised and answered in this original IND submission, but honestly, I don't remember it being a topic that we spent a great deal of time trying to address.
All right. Thank you, Kathy. There's some questions in the chats as well, so I'll get some of those. So one comment/question says, we've often encountered regulator hesitancy when trying to bring clinical trials into pediatrics without preceding adult trials, even when adult pathogenesis does not extrapolate well to pediatrics. Did you encounter any similar challenges? If so, how did you address it?
Well, we certainly did encounter that. And I failed to say, and I need to say, for everything that we did, the first 3 participants were always adults. And then the DSMB reviewed so for phase 1, for the 2 phase 1 trials, and for phase 3, the initial participants were adults, the first 3. And then the DSMB reviewed those data, and only if that showed safety did we begin to enroll pediatric individuals. And yes, it was the subject of an entire RAC session for us. The pros and cons of including children in the trial at all. And, I think that all of that was appropriate. And, but I also think that we had, as is often the case for a number of genetic diseases, we had compelling data that children were the people perhaps most likely to benefit from the treatment. And then I'm referring to the dog data. And, so I think that we had to make the arguments and provide the data. And then we had that additional safeguard of at every dose and the dose escalation and so forth, the initial patients were adults.
Understood. Thanks, Kathy. More questions, if you're okay with taking a few more.
Yeah, I think I'm fine. I'm just gonna, I don't always get that right, so let me quickly check the schedule here. Yes, I have a few more minutes.
Sure. Okay, we'll take maybe 1 or 2 more questions and then we'll wrap it up. Which AAV and promoter were used for the trial? What were the findings of biodistribution of AAV?
We used an AAV2 capsid, and the promoter was not RP65 specific. It was essentially a housekeeping promoter. It was a chicken beta actin promoter.
Perfect. Thank you. Final question.
Sorry. Yeah. With a CMV enhancer, I think. Anyway, yes.
Okay. One more question, and that's it. How well does the mouse model reflect the natural dog model and the human condition?
The mouse model results in very early blindness. And I don't know whether that is exactly proportional to the mouse model that we used in the potency assay. I don't know whether it's exactly proportional to what one sees in humans. I mean, I will say that in humans, you do encounter a range of onset of visual difficulties. For example, there were some children who were still in sighted classrooms as elementary school students, and other children who had been in Braille classrooms from the beginning. There were even sibling pairs where the older child was still in a sighted classroom and the younger child was already in a Braille classroom. So in other words, they're clearly disease modifying genes as well, because you can have the same mutation as 2 siblings did, and yet your disease progresses at a different rate. An important point, I think, in this in this disease is that we could not make an age cutoff. And in the label, there is no age cutoff because there were people with mild mutations who actually got a gain of function after injection in their 40s. And so it really does depend on the condition of the person's retina. And what the label says is that in the judgment of the treating physician, the patient has sufficient numbers of cells to support the activity of the donated gene. And so I do think that's an important point. The dogs are different because they all had the same mutation and they all lost vision, on essentially the same time frame. But, humans aren't like that. There are many, many different mutations described in the RP65 gene.
Thank you so much, Kathy. We're just excited to have you. This was
okay.
excellent.
Well, I apologize for all these difficulties. I have to tell you that I seem to always have trouble with Teams. But
no worries,
anyway.
Kathy. We're also learning to use Teams, so we'll all get better with time. We are very thankful for you to be here on behalf of the AGM team and CATS, as well as all of our listeners in this call. We're very thankful for you to be here.
Okay.
Thank you. And then for everyone else, our next speaker is going to be Dr. Carolyn Bertuzzi, who's a Nobel laureate. She's going to be our Q2 speaker, and that's on June 12th. Stay tuned. Thank you, everyone. You all have a good afternoon. Bye.